You guys caught me getting some of my earwax out. But while we're on the topic of earwax, did you know that we actually have a gene that determines what our earwax is going to be like? It's true, and that's what this video is all about. So we have 23 chromosomes, and on chromosome number 16 is where this earwax gene is found. And this earwax gene determines whether you're going to have dry earwax or wet earwax. So as I said, this gene is found on chromosome 16. So let's say it's right here. It is actually located close to the centromere, so remember the middle part there. So let's say it's right here. We'll call it the earwax gene. Its actual name is ABCC11. So there's a little interesting tidbit for you. So that's our gene. Now remember, alleles are the different forms of a gene. Now our two alleles or forms of a gene in this case is that the earwax could be wet, and that's dominant. So we'll use a capital W to represent that, or it could be dry. So we'll use a lowercase w because that's recessive. So we have the earwax gene, the two alleles or forms are wet and dry. So let's think about someone who is heterozygous for this trait. That means they have one dominant allele, one recessive allele. So in their body cells, then, this homologous pair, one of these would have the dominant allele, the other one of these would have the recessive allele on it. Okay, so they got one from mom, one from dad. Then, in the cell cycle, that DNA is going to be copied. So that's why we usually think of chromosomes looking like this, because remember from last semester, during S phase, those get copied. So this would be an S phase. They get copied. Now we have two copies of that dominant allele and two of the recessive allele, okay? And the cell continues on in G2 phase, preparing to divide. Now, last semester we learned about if it went through mitosis and split, but let's talk now about meiosis and review that. So say that body cell went through meiosis. We can remember that at the beginning of meiosis in prophase 1, those homologous chromosomes line up, and this is when crossing over, or they pair up, and this is when crossing over would happen. So remember, we have big W, little w, little w. So let's say these don't, that section doesn't cross over. These stay like that. So then we go through meiosis 1, and the homologous chromosomes are split apart. So we have big W, little w, little w from meiosis 1. Then meiosis 2, those sister chromatids are split apart. And so we end up with, this one has a little w, recessive allele, dominant allele, and dominant allele. I should make that look bigger. So this can help us remember, one, that not all of the gametes that are produced here are identical, right? Some have this dominant allele, some have the recessive allele. And it's important to think about that we don't just have one pair of chromosomes, we have 23 pairs of chromosomes. So the gametes that are produced are going to be, there's going to be a lot of variety between them depending on how those chromosomes line up way back here. So that's the first point to remember. Second point to remember is that this is why parents only pass on one allele, is because of meiosis. So here at the end, let's say this is a man going through meiosis and these are sperm. They only have that one allele instead of both in there. So that's all they're going to pass on. So what if we were to take this heterozygous person we've been talking about and cross them with someone that is homozygous recessive? The first thing that we always want to do when we're doing a Punnett square is write out the parent genotypes. So in this case, our first parent was heterozygous, and the second parent is homozygous recessive. So now we'll put those parent alleles onto the Punnett square. Now, I left these up here because this is where the child is getting these alleles from these gametes, the eggs or the sperm. And so if we look up here, this heterozygous person, they can only give a dominant allele. So we'd put that one here, or a recessive allele. So that's why we split the alleles up on the side of the Punnett square. It's because of meiosis. Then the other parent can only give recessive alleles. So then we bring these into the middle to whichever one they're lined up with. That offspring would be heterozygous. This one's homozygous recessive. Heterozygous, homozygous recessive. And so the inside of the Punnett square here represents the potential offspring. These are the possibilities of the genotypes of these people's children. And so in this case, half of these potential offspring could have wet earwax. Remember, wet was the dominant. The other half, these two, are going to have dry earwax because they have both recessive alleles. So we would say there is a 50% chance offspring would have dry earwax and a 50% chance that the offspring have wet earwax.